to the next talk, for which we have very eminent gastroenterologists joining in as chairpersons. We have Professor Amit Dutta, who is a professor of gastroenterology at CMC Bangalore. And we also have Dr. Sanjay Chandani, who is an assistant professor in Department of Gastroenterology at B.Y. L. Nair Hospital in Mumbai. Over to you, chairpersons, for the next session.
Sanjay, we can't hear you.
Voice is unclear.
Sanjay, you are muted.
Yeah, Dr. Datta, please.
Yeah. So thank you, Dr. Vishal and Dr. Usha Datta, for inviting me for this meeting. It's an excellent meeting. The academic content has been brilliant. And in this, we now move on to the next session. So can I have the speaker slide, please? The first slide of the speaker. Yeah. So the next topic is immunological targets for IBD. Now we know that knowledge of the immunological pathway has led to an explosion of the number of drugs we have for IBD, and it's actually increasing day by day. So we have this very important topic. It will be delivered by Dr. Shrinivas Bishu, who is an assistant professor in Division of Gastroenterology, Department of Medicine at University of Michigan, Ann Arbor. So over to you, Dr. Bishu.
Hi, thanks a lot for this opportunity to talk. So just so everybody knows, I'm Shrinivas Bishu, and my background is obviously clinically IBD, but I do a lot of sort of basic science and translational science with microbiome and immunology. And that's really my area of focus. So today I'm just going to give you guys a rough overview of how these targets are and what the sort of basic biology of IBD is. And I'm trying not to go too deep into the woods, but I'm happy to answer any questions that people have. If you just give me 1 second to pull up my slides here. Okay, everybody see my slides?
Yeah, we can see it. Yeah.
Okay, perfect. Thank you. Okay, so immunologic targets for IBD, and what I was tasked to do was really talk about the biology of IBD and sort of give a broad overview of how drugs work, and maybe what the future holds. So, okay. So just an overview. So first I'm going to build sort of a theoretical framework so we can have a conversation about it. And then we're going to talk about the components of the system. And then we're going to move to the present, which is really biologics and small molecules. And I'm going to skip over methotrexate and Imuran, not because I don't think they're important. It's just for the purpose of sort of understanding these pathways and what the future is. It's easier, quite frankly, to talk about biologics and small molecules. And then I want to touch a little bit on the future, specifically the microbiome, microbial metabolites, the intestinal epithelium. And then I'll end just a touch with fibrosis, because I know that Florian is talking about it later. And if you want to have a graphical presentation, it's on the right there, where we talk about the big picture, the host and the microbiome, and then move on down. And I'm going to focus on T cells, only because, again, it's easy for me to talk to in terms of a model system, okay? All right, so very simply, biology of IBD, we know that changes in the gut microbiome, what we call dysbiosis, is related to a dysregulated host response. And if you think about the entire system as the host and all the gut microbiome, that organism has a name, and it's called a meta-organism. And you can think about IBD as a dysregulated response of both the human and the gut microbiota to environmental stimuli. So that's kind of like if you were an evolutionary biologist, you would say that that's just a bad reaction to environmental stimuli. Now, when we talk about the microbiome, people typically talk about bacteria, but it's really important to remember that there's also fungi. Fungi also make up depending on various studies, 2 to sort of 10% of the microbiome. There's actually also viruses in there that infect bacteria. And there's also the stuff that fungi and bacteria make, which are the metabolites. On the host side, there are tons of different sort of cells that are involved, but the important pieces, the big pieces, are the intestinal lining itself, so the epithelial cells, And then kind of two big classes of immune cells, which are antigen presenting cells, and I'll get into that in the next slide, and T cells, and their interaction is critical to IBD, and they're represented by the bottom cell types on this figure here. And then you have a variety of cells that are called stromal cells, which is just all the other stuff in there, and they're going to be really important for fibrosis. So let's build like a really simple model. This is a figure that I developed just for this talk, and it's just a really, really basic system. So we know that inflammation and dysbiosis somehow interact to basically cause IBD or present as IBD. And these little black things here are the bacteria. The blue squiggly lines is the intestinal epithelium. So you have a break in the intestinal epithelium, and then what's normally contained, the bacteria and the fungi sort of cross over where they're not supposed to be. They end up activating groups of cells. In particular, they end up activating cells called antigen presenting cells, or APCs for short. Those APCs, as we'll talk about in the next slide, engage a special class of cells called T cells. And bottom line is they both perpetuate inflammation by producing stuff downstream. So that's kind of like a working model. And the major problem with T cells and antigen presenting cells and things like that is they stick around for a really long time. And this is how vaccines work, actually. So they stick around and they have a memory for these systems. So T cells and antigen presenting cells. So what happens is you have to think about your body in terms of three compartments. You have your tissues. So in our case, it's going to be the intestine. Then you have lymph nodes and you have blood. Now within lymph nodes, what happens is microbes interact with antigen presenting cells, and then antigen presenting cells will engage T cells. That will start a process, what's called T cell education, and that depends on specific cytokines. In this particular case, I've highlighted IL-1 beta and IL-23 because, as you'll see later, those are targets for IBD. And then those T cells become educated into differentiated T cells that are characterized by specific cytokines and specific sort of transcription factors, which are basically the brains of the cell. Once this happens in a lymph node, they get out into the blood and they traffic. And they traffic to peripheral tissues where they perform what are called effector functions. In this simple diagram, they're going to traffic out of the lymph nodes, go to the blood, go into the tissue, and basically clean up the microbiota that are not supposed to be there. So in this, very rough and simple sense, we treat IBD by simply targeting these steps. Okay, so how do we do that? So I'm going to blow it up on T cells. Now, T cells, in this particular case, this group of T cells will require IL-23, the cytokine IL-23, to develop. They will require intracellular signaling to turn on their processes, and that's where JAK or Janus kinase inhibitors come in. And then they need to traffic, and the way they traffic is they use these special receptors, in this particular case, alpha-4 beta-7. And then finally, they need to sort of do their job. And their job is going to be to produce TNF to eradicate bacteria or pathogens or whatever it is. So one really simple way we treat IBD is just by targeting each of these steps. So we impair their formation by blocking IL-23. And an example of that is ustekinumab. We impair intracellular processes by targeting intracellular signaling. And an example of that is tofacitinib, which inhibits this Janus kinase chemical. We can block their trafficking by using things like vedolizumab. And I think Takeda is a sponsor here, so they make vedolizumab. And then finally, we can eradicate their function with anti-TNFs. Now, this is a very simplified model. So if people have any questions about details, I'm happy to answer them. But in a broad philosophical sense, you just want to try to block each of these steps. Okay, so what's available? In the United States and the Western world, these are the medications that are frequently used in terms of biologics and what are called small molecule inhibitors. So for ulcerative colitis, you have anti-TNFs and you have anti-integrins, which block trafficking. You have JAK inhibitors, which block intracellular signaling. And then you have what are called anti-cytokine therapies. Now, many of these medications are similar between Crohn's and ulcerative colitis, but they're not the same. So for example, tofacitinib, which is a JAK inhibitor, is not currently approved for Crohn's disease. And the data is a little mixed on how well they're actually going to work in Crohn's disease. In addition, not all the medications of the same class will work equally well. So Humira, which is adalimumab, and Remicade, which is infliximab, and now we have biosimilars, probably aren't equivalent drugs. So there's pharmacologic properties of the drugs themselves, even if you stop the same process. And then the one other kind of take-home point I wanted to have is these drugs have different time to onset of action. So because they work in different ways, some drugs are going to be faster than others. And the classic group of drugs that takes a little time to act are these anti-integrins. So for whatever reason, they'll take a little longer to be effective compared with a JAK inhibitor or an anti-TNF. So what's the future? Now, in terms of anti-cytokine therapies, and they impair cell generation, there's a lot of these coming out, and they seem to be fairly effective. I've just listed some of them. They're all going through either phase 2 or phase 3 clinical trials in the United States and Europe. This class of drug I haven't talked about yet, but they also impair cell trafficking, and they're a pill form, and they are also going through phase 2 and phase 3 drugs. They have some issues with them, and then the JAK inhibitors, again, like more drugs that are coming through in terms of the JAK inhibitors that impair intracellular processes. But just think, you have an intestinal barrier, bacteria come across the intestinal barrier and activate your cells. And a simple way to think about how are these drugs working in terms of IBD is they just stop steps along that process. So you can stop your pathogenic cell from forming, you can stop it from moving someplace, and then you can stop what's going on inside the cell. And that's what these agents do. Okay, what about future targets? So those are all present targets, but there's a broad future. So let's separate, all these components. We can think about the microbes, right? We can change what's there. We can think about the metabolites that the microbes make and see if we can modify that. And the classic example of that are short chain fatty acids, which are really important for intestinal epithelial cell healing. We can target the epithelial cells themselves and try to help them heal better. And then lastly, we can target stromal cells, all the other cells inside the intestine, to really reduce fibrosis, which is a big problem in Crohn's disease once it's formed. And I think Florian is going to talk about that as well later. So coming back to our simple model, the classic example or the easiest example to understand in terms of changing the microbes is fecal microbiota transplant, so FMT. We know that the microbes are linked with IBD, don't exactly know how, but we know they're linked. So the simple thing is, can you change the microbes? And actually C. diff and FMT for C. diff that was given in the previous talk is a very good example of that. This is not perfect, and we clearly have not figured out how to do this. So two of the most obvious problems are which bacteria, we don't know, and what mix of bacteria, because you have to view the human intestine not a single isolated species, but it's actually microbial ecology. So one group of bacteria impacts another, and you can think of C. diff that way. So the traditional risk factors for C. diff are a reduction in diversity of bacteria, either by antibiotics or due to IBD. And then C. diff, which itself is not a very bad pathogen or not a very strong bacteria, so to speak, kind of takes over. So that's an example of that ecology. So we have not figured all of this out yet in terms of FMT, but I think the future is going to involve how do we know which bacteria help and which donors help which recipients. So that's really one easy way to sort of change this, and there are different ways to do this. Okay, what about metabolites, right? So microbes make beneficial things. As we talked earlier, short chain fatty acids are a classic example of that. But there are other beneficial things that microbes make. And while you're talking about microbes, the viruses that infect the bacteria can actually be utilized, and they're called phages, bacteriophages. So that may be a thing in the future too. There's a lot of ways to fix microbial metabolites. So diet, in particular high fiber, prebiotics, probiotics, just giving the metabolite itself. So people are actively investigating these issues. But again, the issue is which metabolites. And if you're talking about purely dietary changes, most studies of diet that have been done are actually pretty not great results. And one of the issues is people struggle to change a diet. And the other issue is that diets are just super, super complicated. So we don't really know how to modify this, but that's also a future direction. Okay, what about epithelial cells? So we know that the damage is happening at the level of an epithelial cell. We can try to fix it faster. And there are really well-known cytokine families that help heal epithelial cells. There are lots of problems with this. One of the issues is a lot of the initial trials, in particular with IL-10, which is really well known in animal models to heal epithelial cells, doesn't seem to work. And that could be because we're not delivering enough drug, or we just don't know the right way to get it there. But the other problem is they may actually promote cancer, because if you have intestinal epithelial cells and you're regenerating them, usually you're messing with the stem cells, and the stem cells turn on genes that just can promote cancer. So that's a future thing I think will happen, but we'll have to work on this. And then lastly, stromal cells, right? So once you have damage, in particular, once you have fibrosis in IBD, is there a way to reverse it? Or is there a way to sort of reduce the activity of these stromal cells, which is this yellow guy hanging out in the corner in the figure here, that can prevent people from having surgery? I think Florian is going to talk about that later, so I won't say much more about that. But with that, I'm going to kind of end it. This is just a graphic of sort of how involved that whole process is, in particular for fibrosis. So there's a lot of players. So just to kind of summarize, we can target the host, specifically how these cells are formed, maintained, traffic, or function. We can target the microbiome, specifically the bacteria, maybe even the fungi, or what they make. We can target epithelial cells and try to help them heal. And we can target stromal cells to improve healing and reverse fibrosis. And I think the other kind of thing you're going to see in the future is there's not really a reason we can't mix and match. So it may be a future in IBD where you have a particularly refractory patient or a group of refractory patients who, say, for example, get an anti-TNF and something to heal the epithelium, or get a JAK inhibitor and something to improve fibrosis, in addition with microbiome modification with diet. So people talk about like dual therapy as an impending concept, and I think it is a concept, but it's important to remember that dual therapy doesn't just apply to medications. It applies to multiple approaches to sort of deal with this problem. I think that's it. So thank you for your time. Anybody have questions?
Yeah, so thank you, Dr. Shrinivas. That was a wonderful presentation, a good overview of what is on the horizon for IBD. So I'll start with one question. Now, you see, there are a lot of developments happening. And, when you talk about drugs, the response is different in different. Some patients respond to one group of drugs, some respond to one group of drugs, So is there something, there is a difference in biology between patients, but is there something in the pipeline to decide, something in the pathway of a patient to say, okay, the patient may benefit from this particular drug? But I mean, it's a precision medicine, because that's something also which we have to look at.
Yeah, no, that's a really good question. So a lot of people are working on that. The short answer to your question is we have not figured it out yet. That's the short answer. Okay. But the long answer is, if you look at like the things that make people susceptible to IBD, and this is only a fraction of the population. Most people with IBD have no known reason why they got it, but about 20% have like genes, and there's actually over 200 genes that are susceptible alleles to IBD. So I think instead of thinking of IBD as one or two diseases, it's probably better to think about it as like a giant heterogeneous pool of diseases, and what you see is an end manifestation. But a lot of people are working on it from clinical data, like what clinical data predict response to one agent or another. A lot of people are working on it from microbiome data, immunologic data. And I'll tell you that almost certainly anytime a clinical trial is done, the pharmaceutical companies scoop up all this metadata, and they haven't figured it out either. But I think that's sort of the holy grail for the future.
That's an important area which should be looked at apart from the different drugs. So Dr. Sanjay, if any other question, you want to take it, Dr. Sanjay, please? Question. I think okay, there's a problem with his audio. So I'll ask this one question from the audience. They say that, can immunohistochemistry of tissue to assess the predominance of TNF, IL-23, integrin, etc., help in choosing the treatment modality? Somewhat like what I asked you. So basically, can immunohistochemistry help us decide what therapy to offer to a patient?
Yeah, so that's an interesting question. To date, there is one paper one high quality paper that is published about TNF staining, and they used actually flow cytometry based staining, kind of immunohistochemistry, which was published in Nature Medicine, which is like a top shelf journal. That is the single only paper that has been able to do this. To answer that particular question in a broad scientific sense, if you stain for anti-TNF in the intestine, there's just a ton of it. There's like so much of it. And I do this routinely. You can't tell based on that. It's kind of the.
Yeah, I mean, also it's not a homogeneous inflammation. Even if you say target, even a biopsy, you'll be just sampling some areas, isn't it? So really, I mean,
correct.
That's other thing. So it's like, as of now, it's difficult just by histopathology. Read something more than that, actually. Yeah. So, Dr. Sanjay, over to you. Yeah. I think there is some problem with Dr. Sanjay. So I think with this, we'll wind up this session. And thank you, Dr. Shrinivas, again for your wonderful presentation. So with that, we come to the end of this session. Thank you. Yeah.
Thank you.
Thank you, speaker and the chairpersons. With this, we'll now